Thank you very much and please sit comfortably. Ladies and gentlemen, our next agenda is speeches. The first is welcoming speech from Chairwoman of the Committee Research and Review Article Competition 2021. Yuandani S-Farm Master of Science, PhD Apotheker. Time is yours.
Thank you to the MC. Assalamualaikum warahmatullahi wabarakatuh and very good morning.
Waalaikumsalam warahmatullahi wabarakatuh.
First of all, let us praise to the Almighty Allah Subhanahu wa Ta'ala for His blessing, so we are able to attend this valuable event today. Salawat to the Prophet Muhammad Sallallahu Alaihi Wasallam. The Honorable Rector of Universitas Sumatera Utara, Dr. Murianto Amin, Vice Rector 3 of Universitas Sumatera Utara, Profesor Dr. Apoteker Poppy Angelisa Hasibuan, Dean of Faculty of Pharmacy Universitas Sumatera Utara, Dr. Apoteker Hairunisa, Head of Research Unit of Universitas Sumatera Utara, Profesor Dr. Robert Sibarani, Keynote Speaker, Dr. Apoteker Emil Salim, Judges, participants, and Committee of Research and Review Article Competition. On behalf of the Organizing Committee Chair, it is a great pleasure and honor to extend to you all a warm welcome to the Research and Review Article Competition, RRAC 2021. This Research and Review Article Competition held by the Master and Doctorate of Pharmaceutical Science Study Program, Faculty of Pharmacy, Universitas Sumatera Utara, brings out the theme of innovative findings to promote better quality of life in the future. It is a pleasure to share you that this event is initiated by former Master and Doctorate of Pharmaceutical Science Study Program, Professor Dr. Apoteker Urip Harhab. In this year, the participant of competition is an active student from master or doctoral program of Universitas Sumatera Utara. Hopefully, next year, this event can be continued with more master or doctoral students from various universities in Indonesia. We do hope this event can motivate and facilitate master and doctoral students to do high quality research, prepare manuscript to be submitted in reputable international or accredited national journals, as well as present their works in the form of poster and oral presentation. To the keynote speaker, I would like to say thank you for sharing knowledge and experiences to all of us. I will also thank to the judges for the willingness to evaluate and give suggestion to the presenters. To the all participants, thank you for joining us today. Although we only meet virtually, I hope it doesn't decline our spirit to gain knowledge. In this competition, we have 34 presenters. This includes 22 oral presenters and 12 poster presenters who will present their research and review articles. The selected article will be processed to be published in Talenta Conference Series, Tropical Medicines. Finally, many thanks and appreciation I convey to all organizing committees who have cooperated and worked hardly to make this event successful. May Allah Subhanahu wa Ta'ala keeps blessing to all of us and to this event. Wabillahi taufiq walidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, the Honorable Dr. Yuandani. Ladies and gentlemen, now we are going to listen welcoming speech from Dean Faculty of Pharmacy, Universitas Sumatera Utara, Hairunisa M.Com, PhD Apotheker. Please welcome the Honorable Dr. Hairunisa. Time is yours.
Thank you, Rima. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. The Honorable Rector of Universitas Sumatera Utara, Dr. Murianto Amin SSOS MSI, Third Vice Rector of Universitas Sumatera Utara, Profesor Dr. Apotheker Poppy Angelisa Jaitun Hasibuan MSI, Head of Research Unit of Universitas Sumatera Utara, Profesor Dr. Robert Sibarani, Keynote Speaker, Apotheker Emil Salim, S.Pharm, MSc, PhD, judges, and all participants, welcome to the Research and Review Article Competition 2021. All praises to Allah Subhanahu wa Ta'ala. With Allah blessing today, we can meet and gather in this event. Also, salawat and peace be upon Rasulullah Sallallahu Alaihi Wasallam. First of all, I would like to congratulate the organizing committee for successfully organizing the Research and Review Article Competition RRAC 2021. This event is conducted in accordance to support the achievement of faculty performance indicators, particularly for the student performance. This year's event has term of innovative findings to promote better quality of life in the future. Findings in research, particularly in pharmacy field, are important things to obtain to solve the health problems. Students are encouraged to do a good research in order to get high quality result. By joining this event, we do hope that all of the participants can share their innovative findings, compete each other to get the most prominent winner. In this event, we also believe students can sharpen their critical thinking, improve their ability in delivering presentation and English language implementation, and also increase their self-confidence. As the Dean of Faculty of Pharmacy, Universitas Sumatera Utara, I would like to encourage all master and doctor students to take this opportunity to show their best performance. I am sure that with great student performance will contribute to the faculty and university performance. Finally, I wish you success in the research and review article competition 2021 and good luck. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, the Honorable Dr. Hairunisa. Ladies and gentlemen, the next is speech followed by opening ceremony by Vice Rector 3, Universitas Sumatera Utara, Dr. Poppy Angelisa Jaitun Hasibuan, Master of Science, Apotheker. Please welcome the Honorable Dr. Poppy. Time is yours.
Thank you. Ladies and gentlemen, please allow me to read the welcoming speech of Rektor Universitas Sumatera Utara. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Shalom, Om Swastiastu, Namo Buddhaya, and keep healthy for all of us. Honorable invited speaker, Mr. Emil Salim, MSc, PhD Apotheker, Dean of Faculty of Pharmacy, Vice Dean of Faculty of Pharmacy, lecturers, and all participants. Praise the Lord, God Almighty, Allah Subhanahu wa Ta'ala, for His mercy, compassion, and blessing, so that we could virtually gather here this morning in the very event of Research and Review Article Competition 2021. Ladies and gentlemen, honorable conference participants, first of all, I would like to congratulate Faculty of Pharmacy Universitas Sumatera Utara especially the organizing committee of the Research and Review Article Competition 2021, who has successfully conducted a virtual event of competition. Scientific discovery is essential to the development of science. With new discoveries, many problems can be solved, including problems in the field of pharmacy and health. With the theme of innovative findings to promote better quality of life in the future, I hope this event can compile all great solutions to overcome problems in pharmaceutical and clinical field. I am delighted to welcome all of master and doctoral students who have joined Research and Review Article Competition 2021. I hope this event will build strong character of the students in academic and personality area. A competition will directly force us to cross existing limitations. It will transform us to be the best part of ourselves. Aligned with USU tagline, transformation towards the ultimate, I believe that all of the students participated in this event will have ultimate performance. Last but not least, I would like to take this opportunity to express my deepest gratitude to the great speaker and members of the organizing committees for their outstanding preparations and arrangement for this event, as well as all the participants who have made this event successful. Finally, I wish you all have a great moment and best of luck in this competition. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, the Honorable Dr. Poppy. Ladies and gentlemen, now we will take a picture for a while. So please sit up on your best position. Okay, we will screen. 1, 2, 3. Okay, now we will go to the next screens. Please your camera. One, two, three. Okay, now we will go to the last screen. 1, 2, 3. Okay, thank you very much, and please sit up comfortably. Well, ladies and gentlemen, we are going to the next agenda. The next agenda is the lecture with the topic performing toxin monolysine from the entomopathogenic bacterium Pseudomonas entomophila, a molecular and functional analysis. With the speaker, Dr. Emil Salim. This session will be moderated by our moderator, Mrs. Ade Srirohani. Before that, we would like to introduce our moderator. Mrs. Ade Srirohani is the lecturer of Faculty of Pharmacy, Universitas Sumatera Utara. Mrs. Ade Srirohani got her bachelor degree at Faculty of Pharmacy, Universitas Sumatera Utara, as well her apothecary and master degree. A research area of interest is pharmacology pharmacy. Well, without waiting any longer, to open the session, please welcome Mrs. Ade Sri Rohani.
Thank you very much to the Master of Ceremony for a nice introduction. Good morning, everyone, and Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to say thanks for having me as a moderator today, and also thank you very much to the Research and Review Article Competition 2021 Committee for organizing this amazing event, and to all the admirable participants. Welcome to the keynote speech sessions. I am Ade Sri Rohani, will be guiding you in this session. And before we begin, let me remind you for one thing, this session is divided into two parts. The first one is the keynote speech sessions. We have for about 60 minutes, and we will accompany by our speakers, Dr. Emil Salim. And in the second part, we have a Q&A sessions, and it takes for about 30 minutes. And I hope you can enjoy and give focus on our keynote speech later. And if you have questions, you can submit it in the chat rooms. Okay, dear all the participants, let me introduce our speakers today. We will enjoy the presentation from Dr. Emil Salim. Dr. Emil Salim is a secretary of a master and doctoral degree of Faculty of Pharmacy, Universitas Sumatera Utara. Assalamualaikum, Dr. Emil Salim.
Waalaikumsalam warahmatullahi wabarakatuh, Mrs. Ade.
How are you today?
Fine, thank you.
Alhamdulillah, thank you. Okay, ladies and gentlemen, Dr. Emil Salim is a bachelor degree from Universitas Sumatera Utara and also in apothecary profession program. And the master degree from University Kebangsaan Malaysia, Kuala Lumpur, Malaysia. And the doctoral degree from Kanazawa University, Kanazawa, Japan. And Dr. Emil Salim has a teaching subjects and research interest area in immunology and pharmacology field. And he also has many poster and oral presentations and publications in reputable international journals, including about monalizin as a forming toxin. Okay, the other participant, without belonging for a long time, perhaps we can make a start the keynote speech session with the topic for forming toxin monalizin from the entomopathogenic bacterium Pseudomonas entomophila, a molecular and functional analysis. It's my pleasure to welcome our honorable speaker. Please welcome Dr. Emil Salim. We are so good to have you here, and the time is yours.
Thank you, Mrs. Ade. Please let me share my screen. Wait a minute. Wait a minute.
Okay, maybe meanwhile we are waiting for preparing our speaker's presentation. Maybe I would like to say about a little information about our topic today about performing of monolysine. That this topic is based on our speaker's research in Japan, and maybe it's a interesting topic for us. Yes. Because it's a new topic for us, especially in Universitas Sumatera Utara, and maybe it's a new chance for us to take a new research about the monolysine or about the Pseudomonas entomophila from the Drosophila melanogaster, maybe. Okay, ladies and gentlemen. Yes. Okay, let's enjoy the presentation from our speakers today, Dr. Emma Salim. Please welcome Dr. Emma Salim.
Okay, thank you, Mrs. Ade Rohani. I'm sorry for taking time to prepare the presentation. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable Rector of Universitas Sumatera Utara, Dr. Murianto Amin, SOS, Vice Rector 3 of Universitas Sumatera Utara, Professor Dr. Apoteker Poppy Angelisa Zaitun Hasibuan, SSC Master of Science, Dean of Faculty of Pharmacy of Universitas Sumatera Utara, Apoteker Khairunisa, SSC MPharm, PhD, respected Vice Deans, Head of Research Unit of Universitas Sumatera Utara, Professor Dr. Robert Sibarani, participants and Committee of Research and Review Article Competition, judges, teachers, and professors. Praise be to Allah Subhanahu wa Ta'ala, and salawat and peace to Rasulullah Sallallahu Alaihi Wasallam. Thank you very much to the organizers for giving me this opportunity to present our studies in this exciting event. Today, I'm going to talk about monolysine, a pore-forming toxin from Pseudomonas entomophila. The study was conducted in Japan at Kanazawa University. So here is our city, Medan, and then if you want to travel to Japan, there are two possibilities. First, from Medan to Jakarta, and then from Jakarta directly to Osaka or Tokyo. Then from Osaka or Tokyo, we can go to Kanazawa. And also, we can also go to Kuala Lumpur. And then from Kuala Lumpur, again, we can go to Osaka or Tokyo. It takes around 6 hours from Indonesia to Japan, and then from Tokyo to the city that I studied about this field is around 6 hours as well. So this is the figures of my city when I was there. This is the gate of Kanazawa Station. So this is very famous, one of the best station, very beautiful station in the world. Also, if you come to Japan, of course, you can experience 4 kinds of seasons. Like this, this is sakura. And then during fall, you can experience many flowers and leaves fall. And during winter, especially in my city, it is very cold during winter, and the snow is very high. And this is the figure during summer. So I hope you all can come and visit Japan. So this is the figure of Kanazawa University. Here is my building. In here, I perform research about many things, especially about the study monolysine. So this is one of the conference that I attended before COVID-19. Of course, this is me. Maybe all of you already know. And this is my supervisors, quite young. This is Associate Professor Takayuki Qureshi, and this is his supervisor, Professor Bruno Lemaitre. If you know about toll pathway, toll receptors, he found this one, and then this work got Nobel Prize. I would like to divide my presentation into introduction, and then background and general purpose, results that contain purification of monalizin from Pseudomonas entomophila, electrophysiological characterization of monalizin as a pore-forming toxin, and then we use HSIFM or high-speed atomic force microscopy to study the structure of monalizin. So this HS atomic force microscopy is very advanced microscope that is developed by one of the researcher in Kanazawa University where I study. Then we also observe the movement of monalizin using this microscope. And conclusion. Okay, first let me start from introduction, background, and general purpose of the study. Because I will use I use Drosophila melanogaster or fruit fly in the study. I would like to introduce you about this animal. There are many advantages of using Drosophila melanogaster or fruit fly. They are very easy and cheap to keep. And a fruit fly generation takes about 10 days. Thus, fly research progress rapidly, and pedigrees over several generations can be easily planned and monitored. And then the fly genome is a lower redundancy than in higher organisms. 75% of non-human disease genes that have a recognizable match in the genome of fruit flies. So there are disadvantages makes sense that we can use this animal model to study many biological phenomena. Not only to study molecular phenomena, we can also use this animal to screen drug effects. This fruit flies is the animal behind these 10 Nobel laureates, such as Morgan for its study about the role of chromosome in heredity, Muller about mutation by X radiation, Lewis, Nielsen, Forhard, and Viscos to study the genetic control of early embryonic development, Axel for its study for other receptor, and this is as I mentioned before, Hoffman together with Bruno Lemaitre, he found toll pathway in the activation of innate immunity. And recently, Hall, Rosbach, and Young found molecular mechanism controlling the circadian rhythm. This is the introduction about Drosophila melanogaster. You can see that Drosophila melanogaster is one of the powerful model organisms to study various molecular mechanisms. And next, let me introduce you about Pseudomonas entomophila. This is the figure of Pseudomonas entomophila. Pseudomonas entomophila is a gram-negative bacterium isolated from Drosophila melanogaster in Guadeloupe, France. So quite far we got this fly. Pseudomonas is unique among Pseudomonas species in being able to naturally infect and kill insects upon ingestion. Pseudomonas entomophila is one of the most important models for the study of insect microbe interactions. So of course, from the study insect and microbe interactions, we can get insight how human and microbe interact. Upon ingestion, this strain was able to activate a systemic immune response in both Drosophila larvae and adults. These bacteria are highly pathogenic to Drosophila and other insects from at least 3 different orders. Oral infections with high doses of this bacterium cause massive destruction to the Drosophila gut epithelium. Thus, Pseudomonas entomophila has a potentially wide host range and making it a promising model for the study of host pathogen interactions and the development of biopesticides against insect pests. This bacteria cause damage to gut epithelial cells by activating Drosophila duoxoxidase, leading to the release of reactive oxygen species that can cause damage to the epithelial cells. Moreover, Pseudomonas entomophila also produce pore-forming toxin or PFT that can damage intestinal cells. Pseudomonas entomophila produce proform protein, what we call promonolysine, that can be activated by alkaline protease A to become active monolysine. Active monolysine then insert to the epithelial cells in the gut, then make pore. After making pore, the cells become leakage and broken. Cytotoxic activity and the structure of recombinant monolysine has been studied. I would like to introduce you what is recombinant monolysine. This is not the natural monolysine. So the scientists design DNA and then insert it into the plasmid of E. coli. Then E. coli produce the protein what we want. So the previous study already done to study the toxicity and the structure of recombinant monolysine from E. coli. And then they found that this recombinant monolysine contain 9 subunits. However, no study has been performed on the endogenous monolysine or natural monolysine produced by Pseudomonas entomophila. This study could provide more precise insight of monolysine function rather than using the recombinant monolysine generated by E. coli that may have different intracellular environment that may produce different subunit composition of protein, maybe not 9 subunit, and could influence the structural and functional features of monolysine. Thus, the study on characterization and function of endogenous monolysine is very important. This study may be useful as tool to explore the interaction of hosts and bacteria, and also to develop biopesticides and also biosensors. To start this study, first we purified endogenous monolysine from Pseudomonas entomophila. First, I would like to show you our previous study that show Pseudomonas entomophila extract that prepared by sonication of bacteria cells with detergent, followed by membrane filtration, kill DCY1 mutant adult survival. So this is also fly or Drosophila, but this time is not wild type, not normal one, but this is mutant one. This fly contain the mutation of DCY1 gene that code DCY or drosocrystalline protein. This protein is important for physical barrier formation in Drosophila digestive tract. So let's have a look to this figure, so this figure x-axis is time post-infection from 0 to 40 hours, and y-axis is adult survival in percent. As we can see in this figure, showing that the survival of DCY1 mutant fed with Pseudomonas entomophylla extract was decreased significantly compared to that of control. In other words, I would like to say that Pseudomonas entomophylla extract killed DCY1 mutant flies. So this figure indicates that Pseudomonas entomophylla extract may contain toxins. Thus, this extract can be used as a source for endogenous monalysine purification. For purification, first we examine Pseudomonas entomophylla extract toxicity on Drosophila S2 cells by incubating S2 cells with Pseudomonas entomophylla extract for 12 hours and then observe the viability of cells under microscope. I would like to introduce you what is S2 cells. S2 cells is cells that derive from Drosophila melanogaster. As we can see in this figure, shows that PBS and Pseudomonas entomophylla extract from Pseudomonas entomophylla monalysine mutant strain didn't disrupt the cell membrane. So we can see here, this is the figure of normal cells, and also the extract that contain the mutation in monalysine also shows normality. However, Pseudomonas entomophylla extract from Pseudomonas entomophylla wild type showed toxicity on S2 cells by damaging the normal form of S2 cells membrane. I think it is very clear comparing this figure to this both figure, because this figure, the shape of the cells not looks normal again like this one. This data indicated that Pseudomonas entomophylla extract was toxic to the cells. We also performed cell viability assay using cell titer glow assay. In the cell titer glow assay, live cells were indicated by the existence of ATP. This ATP will interact with beta-luciferase and luciferase enzyme, resulting luminescence. Luminescence is proportional to the number of viable cells. Similar to the observation by microscope, cell titer glow results show that Pseudomonas entomophylla extract reduced S2 cells viability significantly compared to that of PBS and Pseudomonas entomophylla extract from monalysine mutant strain. These results indicated that Pseudomonas entomophylla extract contains monalysine toxin and can be used for the purification of endogenous monalysine. Furthermore, this viability assay was used to find toxic fractions in every purification step. Next, we perform monalysine purification by these 5 steps. First, we prepared Pseudomonas entomophylla totalized, followed by ammonium sulfate precipitation. Then we perform anion exchange chromatography. Again, we perform ammonium sulfate precipitation. In the end, we carried out gel filtration chromatography. First, I will explain about the preparation of Pseudomonas entomophylla totalized. 2-liter bacteria culture was prepared, and then we perform centrifugation. The pellet were washed with PBS and lysed with PBS chaps combined with sonication. Next, we perform filtration, and again, we perform centrifugation, and then supernatant was subjected to dialysis with PBS, and then we found totalized, or we call fraction 1. After this step, we obtain 3.5 milligram of total protein from 2 liter of Pseudomonas entomophila culture with specific activity 8.7 10 to the power of 2. Next, we perform ammonium sulfate precipitation to reduce extract volume. Totalized that we found from the previous step was precipitated using ammonium sulfate 25%, then 50%. The pellet then diluted with 20 millimolar hydrochloride pH 8 and dialyzed with the same buffer. Then we obtain dialysate, what we call fraction 2. After this step, the total protein and specific activity were reduced. Then we continue to the next step, anion exchange chromatography. We use high trap QHP column. First columns was subjected to pre-equilibration in 20 millimolar hydrochloride pH 8. Then sample was added. Negatively charged protein will be bind to column. Next, this protein was eluted by a linear gradient of 0 to 1 molar sodium chloride. Subfractions were collected at different time points and subjected to toxicity assay. Then toxic fraction were collected and mixed. So this is the result of anion exchange chromatography and chromatogram, and also the toxicity assay of the fractions. The above figure showed the chromatogram from anion exchange chromatography, and this is the toxicity results that we got from the cell titer glow assay. The results show that the elution 4 until 8 were toxic to S2 cells. As we can see from the control, the toxicity of this fraction significantly reduced. Then we collected this elution 4 until 8 and mix. After this step, the specific activity and purification fault were increased. Next, we perform ammonium sulfate precipitation with final concentration 50%. The pellet was dissolved in phosphate buffer pH 7.4. This is fraction 4. After this step, the specific activity and purification fault were increased. The final step, we perform gel filtration chromatography using Superdex 200 column with sodium phosphate buffer containing sodium chloride. Fraction 4 was loaded to the column. Protein will be separated based on their size. The elution then collected in different time points. These subfractions were subjected to toxicity assay. So this is the results of the gel filtration chromatography and toxicity assay. The left panel showed 5 subfractions collected from gel chromatography. After measuring cell viability, we can see that subfraction 1 that corresponded to the highest peak was toxic to the cells. Subfraction 1 showed a sharp single peak around 460 kilodalton and gave a single band with a molecular mass of 30 kilodalton on an SDS page, which is the estimated size of the propome of monolysine monomer. Then we perform MS analysis and confirm that amino acid sequences of the single band correspond to monolysine. After this step, the specific activity and purification fault were increased significantly to 14 faults. We conclude that we succeeded to perform for the first time in the world purification of endogenous promonalysine from the wild type of Pseudomonas entomophylla. Next, we perform promonalysine conversion to monalysine. Promonalysine is considered to undergo proteolytic cleavage by alkaline protease, a protease secreted by Pseudomonas entomophylla, to become active as monalysine. Leon et al. Showed that trypsin cleavage recombinant promonalysine to monalysine, and this trypsin recapitulated or have similar effect as alkaline protease that produced by Pseudomonas entomophylla. Because trypsin is easier to be purchased, then we use this trypsin. Thus, we perform trypsin cleavage on our purified endogenous monalysine to transform promonalysine to monalysine. We perform trypsin treatment 0.2 mg per ml for 10 minutes to activate promonalysine. As we can see in this figure, this is control. We cannot see any band. And then when we, without addition of trypsin, we can see this band around 30 kilodalton. This is promonalysine. After we add trypsin, we can see that promonalysine is converted to 27 kilodalton. So this is already converted to monalysine, not promonalysine anymore. So this 27 kilodalton is similar as previously reported of recombinant monalysine. Next, we tested the toxicity of monalysine on S2 cells. Left panel showed that monalysine from 1.5 microgram per ml reduced cell viability. Right panel showed that LC50 of monalysine was significantly low at around half of promonalysine. This result indicated that trypsin treatment activates promonalysine. Since monalysine, which subjected to trypsin treatment, is more toxic than promonalysine, from hereafter, monalysine is referred as active monalysine. Then, I sought to investigate the mechanism of cell death induced by active monalysine. As introduction, commonly, cell death can be occurred through apoptosis and necrosis. Apoptosis is normal cell death. Initially, cell shrinks and undergo chromatin condensation. Then membrane starts bleeding and form apoptotic bodies that will be cleared by phagocytic cells. Thus, this process will not induce inflammation because the molecule inside of the cells is kept inside and again phagocytoside by phagocytosis cells. Whereas necrosis is abnormal cell death, starting from cell swelling, then cell leakage and lysis, then can release the intracellular molecule that able induce inflammation. To examine the cell death induced by active monalysine, we incubated S2 cells with active monalysine, and then we measure the caspase 3-7 activity, which is marker of apoptosis from 6, 12, 18, 24 hours of incubation. We use cyclohexamide as a positive control for apoptosis, since this compound can induce caspase 3-7 as marker of apoptosis. Then if this marker is induced, we can say that the apoptosis is induced. The results showed that cyclohexamide induced the caspase-3 activity from 6, 12, 18, and 24 hours of incubation. And then, in contrast, active monalizin didn't stimulate caspase-3 activity from all of time of incubations. So we can say that this active monalizin don't kill the cells through apoptosis, but maybe through other mechanism, necrosis. Next, after performing in vitro assay, then we tested the toxicity of purified monalizin in vivo by injecting monalizin into adult hemolymph, then observe the fly survival after 1 hour. Right figure shows that fly survival was reduced significantly upon active monalizin treatment compared to that of promonalizin. This result indicated that active monalizin was toxic to Drosophila in vivo. We also tested toxicity of active monalizin on DCY1 mutant guts by oral feeding active monalizin. Then after 8 hours, gut was dissected out and immunostained. Upper right figure shows that an immunostaining of the septate junction marker dislodge or DLG, which is green color, and nuclear staining indicated by blue color in flies after oral injection without active monalizin. We can see this is the normal structures of the cells in the epithelium in the gut. And then after addition of active monalizin, we can compare from control to this one. We can see that the structure is not organized well anymore. We can see the blue color is not lined like this, but randomly. And also the green color, which is septate junctions, also not in a normal position. So also we perform phospho-3 staining, phosphohistone 3 staining. pH3 is indicator of gut repair after damage. If we found pH3 is increased, so meaning that there is the damage in the gut or in the cells. So also in this experiment, we perform DCY1 feeding by active monalizin. And then after 8 hours, the gut is dissected out, and then we perform pH3 staining. This result showed that the number of pH3 positive cells in the gut were increased after active monalizin treatment, indicating that active monalizin induced damage on epithelial cells of Drosophila. So together, this data showed that active monalizin was toxic in vivo. So as we know before from the previous result that the damage of the tissue or cells is occurred by active monalizin, then because the tissue damage can induce humoral innate immunity activation, we measure antimicrobial peptides, because these antimicrobial peptides is the signs of the activation of humoral innate immunity. If you know, in the mammals, we call these antimicrobial peptides as cytokine. The antimicrobial peptides that I measured were drosomycin. Drosomycin is the readout for the toll pathway. If the toll pathway is activated, then the expression of drosomycin is increased. Also, I measured the expression of diphtherizin. Diphtherizin is the readout of the IMD pathway. Both the toll pathway and the IMD pathway are the main immune pathways in Drosophila. The toll pathway is the pathway to sense gram-positive bacteria and fungi, whereas IMD pathway is the pathway to sense gram-negative bacteria. Also, we measure GNK pathway and JAK-STAT pathway by measuring the expression of Packard and 200A respectively. This pathway also found to be activated by bacteria and also stress in the Drosophila. So that I measure all of these antimicrobial peptides to see whether these pathways are activated upon active monolysine or not. Okay. This is how I perform the measurement of antimicrobial peptides. First, we injected the flies into its hemolymph or thorax by active monolysine, and then after 1 hour, we take the flies and perform RNA extractions and then cDNA synthesis, and then we measure the antimicrobial peptides by qPCR. Then, as we can see here in this figure, after 3 hours and 6 hours and also 20 hours, we can see that PBS didn't stimulate the drosomycin. However, after 12 hours, we can see that active monolysine induced the expression of drosomycin. So we can say that the toll pathway is involved upon induction of active monolysine. And then this is the result of the activation of the MD pathway. As we can see, the diphtherizin expression didn't increase it at any time points. This result indicated that the IMD pathway is not involved. And similar to diphtherizin, Packard was also not induced, indicating that GNK pathway is not involved. Finally, this data showed that active monolysine induced the activation of 200A, indicating that JAK-STAT pathway may involve. So this all results about the humoral activation indicate that the toll and JAK-STAT pathways are induced by active monolysine. Okay, next, we perform electrophysiological characterization of monolysine as a pore-forming toxin. We want to see that whether active monolysine make pore in the membrane. So the method we use in this assay is artificial planar bilayer membrane. In this method, two separated wells are filled with lipid. Then the buffer containing monolysine was dropped to the wells. This will form bilayer membrane. If monolysine able to form pore, then ion will pass through this channel. Pore formation was monitored by recording ion current signals. This figure shows the typical data of current signal upon pore formation. When one pore is formed, the current will increase 1 step. When 2 pores are formed, the current will increase into 2 steps from the beginning. So this is the results of the experiment. The results showed that the current signal was increased upon treated by active monolysine. We obtained a total of 723 stepwise signals specific to nanopores containing proteins in the solution containing, in this case, containing active monolysine. On the other hand, in case of promonolysine, 35 stepwise signals were observed. From this data, we concluded that active monolysine were more vigorously reconstituted into the bilayer and form nanopores within it. We also estimated the pore size of active monolysine. To do this, the current data from the single pore was transformed to current amplitude histogram. X-axis indicates current and Y-axis indicates the number of current. We can see 3 Gaussian peaks in this histogram. Using these 3 peaks, we calculated pore size utilizing this formula. Then we obtain the estimated pore size of active monolysine to be around 0.7 to 1 nanometer. Collectively, this result indicated that the active monolysine is a pore-forming toxin. Next, we perform the study on the structure and dynamic of endogenous monolysine. The use of endogenous monolysine is very important for evaluating innate immune influx and developing biological agents to control insects and as a biological nanopores. Thus, the understanding the detailed molecular function of endogenous monolysine is essential. To this end, we employ high-speed atomic force microscope or HSIFM. Compared to other methods, HSFM is a powerful method to study the structures and dynamics of biomolecules. This is the figure of IFM structures. IFM or atomic force microscope works using a tiny cantilever with a nanoscale tip at the end, which can fill the surface. The interaction between tip and surface provide signal that can be used to generate an image of not just the topography, but also in some cases as an indication of the chemical composition of the structural features in the samples. And let's look to the HSFM. High-speed FM, it is different to this one. This is IFM, and then this is HS, meaning that this is high-speed HSFM, works by increasing the operation speed of FM. As we can see in this video, HSFM works by increasing the operation speed of FM. In this way, as shown in this video, HSFM record walking myosin 5 on actin filament. So this is very sophisticated microscope that can show us the protein movement directly. So like fluorescent microscope also can observe the movement of protein, but not directly because fluorescent microscope use tuck that bind to the protein, then the microscope observe the fluorescence. So this is different to this method that directly observe the movement of the protein. Thus, we use this HSFM to study the structure and dynamic action of monolysine. First, we perform this study together with Professor Noriyuki Kodera from Kanazawa University, who developed this equipment. Promonolysine drop onto mica surface. Yeah. This is mica surface. We drop promonolysine onto mica surface, and then we observe the image using HSFM. The orange color is promonolysine. As shown in this figure, molecules of monalizin with a uniform height covered mica surface. So again, this is mica surface, and then this orange color, all are promonalizin. And we can see similar pattern and similar color intensity. This is showed that all promonalizin have uniform height. And then if we zoom in at smaller scan size, we can see trefoil shaped molecules like this. This one, and then I make the cartoon of this figure like this. This is the looks like trefoil shaped molecules. Importantly, the molecules corresponding to each leaf of the trefoil dissociated form and rebound to a trefoil shaped molecules, indicating that one particle in these molecules is the minimum unit of promonalizin. Okay. Next, we measure the pro and active monalizin height. The upper figure showed promonalizin with the height around 14. The striking difference from the proform, the height of active monalizin was 5.3 nanometer. As we can see here, this is light orange, and then after activation of monalizin, the color become less intense. So we can see this is the scale of the height. So from this, we can compare and we can calculate the height of promonalizin to be 14 nanometer. And then after the trypsin cleavage, promonalizin was converted into active monalizin with lower height to be 5.3 nanometer. This is half, less than half of the promonalizin. This result indicated and consistent with the idea that promonalizin is double stacked disc-like oligomers, dissociate into two disc-shaped oligomers upon proteolytic cleavage. We also measured the diameter of promonalizin and active monalizin, and we can see no significant difference between diameter of pro and active monalizin upon protease activation. Then we observe active monalizin at smaller scan size. We zoom in again using this microscope. Unexpectedly, we found that active monalizin was composed of 8 subunits and form a disc-shaped octamer as illustrated in this right figure. So we can see again in this figure, this is 1, 2, 3, 4, 5, 6, 7, 8. Mers, sorry, this is 8 mers. So from this result, we can say that endogenous monalizin contain 8 mers. And then if we compare to the recombinant monalizin, which is the background of this study, showed that recombinant monalizin contain 9 mer. I would like to say again, recombinant monalizin is not natural monalizin. So we found a new thing that endogenous monalizin is contained of 8 mers. As a resume for the structure of monalizin, promonalizin is a trefoil-shaped complex with one particle is a minimum unit of promonalizin with the height 14 nanometer and diameter of 11 nanometer. Trypsin treatment activate promonalizin into monalizin with diameter similar to the promonalizin, but the height at around 5 nanometer. The HSFM revealed that the structure of monalizin is 8 mers. Next, we visualize real-time dynamics of monalizin insertion into a lipid bilayer using HSFM. In this experiment, active monalizin was dropped onto lipid membrane that attach to polydimethyl siloxane or PDMS surface, then its dynamic actions was recorded. Let's see together this video. This part is artificial lipid bilayer membrane. Okay. So this part is the lipid bilayer membrane. Then active monolysine was added. We can observe that active monolysine, this is active monolysine, was inserted into lipid membrane. This is the insertion of active monolysine into lipid membrane. We observe also that active monolysine was preferentially inserted into the edge of lipid membrane. As we can see in this illustration, this is PDMS surface, and then this is lipid bilayer that contain planar part and then curved part. And we can see in this illustration that monolysine is preferred inserted from the curved parts. So this implies that monolysine may be like to insert to the part of the cells that have curved parts. To sum up, we successfully see the active monolysine in action using HSFM with preferentially inserted into the edge of the lipid membrane. Well, that's all I have for today. Let me now summarize what I talked about. We succeeded to perform for the first time purification of endogenous monolysine from Pseudomonas entomophylla, which is toxic to Drosophila cell line and adult flies. Active monolysine are reconstituted into the lipid bilayer and form nanopores within it. And then we found active monolysine was composed of 8 subunits and form a disc-shaped 8-mers. And our HSFM analysis revealed real-time dynamics of monolysine in action. I would like to thank these researchers. First, from Kanazawa University, Associate Professor Takayuki Kureshi, who supervised me during this experiment, and also Dr. Sauri Nonaka, who performed together with me the purification of monolysine, and then Dr. Akihori, who support for preparing or for providing equipments and reagents, and then Associate Professor Takumi Nisuchi, who performed the MS analysis and MS experiments, and Professor Noriki Kodera. I performed together with him the experiment using HSFM. And also Professor Koki Kamiya and Professor Soji Takuchi from Kanagawa Institute of Industrial Science and Technology and University of Tokyo, who provide the experiments of electrophysiology assay. And also I would like to thank for Dr. Fizan Nainu for teaching for injection of Drosophila, and Rangga Mediota Asri and Dr. Ayu Mashita, who helped for performing RNA extraction and DNA synthesis. And also I would like to thank Professor Bruno Lemaitre, who provide bacteria and also flies. And also thanks to LPDP, who provides scholarship during this study. And also I would like to thank the committee of this event, RRAC, and also to thank GSPS Kakeni, who provide also the fund for this study. Thank you very much. If you have any question, feel free to ask me. Thank you.
Thank you very much to our speaker. What a very interesting presentation. We have informed, the presentation was opened by the introduction of the Japan, and what a beautiful place in Japan, I think. And we have got a lot of insightful from the presentation of our speakers today, especially how the molecular mechanism of action of active monolysine and how its mechanism was found, and also we were informed how the movement protein work by maybe high speed IFM. Okay, dear all the participants, now we are going to a Q&A sessions. We will remind you that if you have questions, you can submit it in the chat room. But before we read some questions here, maybe I want to ask some questions to our speaker, Dr. Emre Salim. We know that there is a long purification. Yes, there is a long purification stage to get effective monolysine, right?
Yes.
I want to know how long you could take the result so that the best purification is was good, and what's the critical point to get the best active monolysine?
Okay. Thank you very much for the nice question. So for purification of monolysine from the first until the end, it takes around 2 weeks. Yes, so there are many critical steps for the purification of protein. Maybe this is very common because we use protein and protein is very vulnerable or easy to degrade with the high temperature. So we need to handle this protein with as we can at low temperature. So we used to use ice so that can maintain the temperature of the monolysine or extract at 4 degrees Celsius. And also if we perform overnight experiments, we perform this experiment in the cold room so that during 12 hours of incubation, this protein still not degraded. So that is the first critical point. And also the other critical points is the molecules that also exist in the cells of this bacteria. This bacteria contains many enzymes that this enzyme can degrade this monolysine. So we need to add protease inhibitors as well in the purification step. That is the second point that we need to be carefully considered. And also we need to be very careful during the collection of the fractions during anion exchange chromatography and also during gel filtration chromatography, so that we can find the fraction that contain high toxicity to the cells. I think that's all what I can say about the purification of monolysine, what we should care about.
Okay, thank you, Dr. Emre Salim. But about the monolysine, maybe is the monolysine is the only one toxin that we can find in the Pseudomonas entomophylla, or there's still another toxin? Maybe we can difference why the monolysine is the best toxin. You choose the toxin monolysine in this research.
Yeah. Well, very nice question. Can you see, Mrs. Ade, this figure?
Yes.
Okay. So to answer your question, let's see this figure. So this is control, only PBS. Okay. And then this is bacteria that have mutation of monolysine gene. So in this bacteria, no monolysine can be produced. Then when compared to what type Pseudomonas entomophylla, you can see here the toxicity of Pseudomonas entomophila wild type is not reduced completely. There are still remain here. Okay, so this is showed. Let's focus on this bar. This is showed there are other molecules that play a role in the killing activity of this from Pseudomonas entomophila. However, we can see why monalizin is the best toxin for Pseudomonas entomophila. We can see here if Pseudomonas entomophila is absent, we can see the reduction is only here. It is around maybe 20%. So you can see if monalizin exists in this bacteria, you can see the reduction of the toxicity until 80%. So we can say that this is the main toxin produced by Pseudomonas entomophila. Of course, there are other toxins, but in this study, we focus on this main toxin.
Wow. Okay. It means that the monalizin has potential mechanism, especially in cell death mechanism. Okay.
Yes,
Thank
yes.
you. Yes. Thank you, Dr. Masali. Maybe we can now we have already some questions here. Maybe I would like to read the first questions from the committee share screens. Yes. The first questions from Mrs. Lia Laila to Dr. Ms. Valim. To purify the recombinant pro-monalizin into monalizin, can we use other enzyme or protease instead of trypsin? And the second question is why the monalizin preferred to attach on the curved part lipid layer membrane? Is there any specific component on the curved part of, or just due to the location of the curve? Maybe to the first question number one. Please, Dr. Emma Salim. Yes.
Okay, thank you, Mrs. Lia Laila. To purify the recombinant promonalizin to monalizin, can we use other enzyme instead of trypsins? So I think we can use other enzyme because originally the enzyme that can cleave recombinant promonalizin is alkaline protease. So meaning that other enzyme also can be used, probably can be used to degrade or to activate or to convert promonalizin into monalizin instead of trypsin. Also, we see we also ever perform experiments, and then at that experiment we don't have trypsin in that culture, and then we can see that monalizin also can be converted from pro into active, and that this experiment indicate maybe other enzyme also released by the cells that can degrade the promonalizin into monalizin. Okay, sorry. So next question. Okay, next question. So why monalizin is preferred to attach to the curved part of bilayer? So this is also interesting question. I can say that maybe this monalizin can detect the shape of the cells. So because cells as phagocytic cells have, if you can see my fingers like this, so maybe here is very curved. So maybe physically this toxin can detect the shape of the cells. Maybe as your information, the mechanism of action of this protein is not studied yet, but there are many mechanisms of how this pore-forming toxin is occurred. First, maybe through the interactions between this protein to the receptor that exists in the cells. Then after the interaction together, they will make from monomer into polymer, then make pore, and then cells become lysed. And also other possibility also in my case in this in monolysin, because from the artificial lipid bilayer, we don't have any receptor on it, but however, this protein also can make pore. So meaning that there is the possibility, the mechanism of action of this protein.
We are very sorry. There are some problems here. Maybe we can continue to the second questions from Mrs. Laila. Please, Dr. Emil Salim, you can continue the answer.
Okay, thank you. Okay. So why monolysin prefer to attach on the curved part lipid bilayer membrane? So this is only speculation because we didn't perform any experiments why this monolysin tend to attach to the curved part of the lipid bilayer. Because to prove this one, we need to perform experiment. So maybe there is the monolysin can detect the shape of the cells. That is the one possibility. And also maybe in the certain part of the cells, there is specific constituent or component of the cells that can detected by the monolysin. So that is what can I answer to the second question from Mrs. Lia Laila.
Okay, thank you, Dr. Emil Salim. Maybe we can use the alkaline protease to replace the trypsin in provide the monolysin from monolysin. And because the membrane cell, there is no the receptor in the membrane cell, maybe the monolysin can detect the shape. Okay, the next questions from Mr. Soni. Thank you, Dr. Emil, for your outstanding presentation. Regarding to your topic, it was refreshing knowledge. And now we know that the Drosophila model can be used to determine or develop mechanism of numerous human genetic disease. My question is, what kind of facility equipment that we need to develop new drug research using Drosophila model at the Faculty of Pharmacy? Maybe it's about the developing of the research about Drosophila melanogaster in our faculty. Please, Dr. Emil Salim.
Okay. Okay. Thank you, Mr. Soni, to raise this interesting question. I hope I can answer this question and maybe heard by the Dean of Faculty of Pharmacy. Then she can help to establish the facility of Drosophila in our faculty. So actually not difficult to establish the facility to grow Drosophila. We only need a small room that have 25 degrees Celsius temperatures. These temperatures is very important to maintain the life of Drosophila. And also it is also good to have 18 degrees Celsius incubators. So if we don't want to use this Drosophila in the maybe we want to keep this Drosophila for a long time, so we can put them at 18 degrees Celsius incubator. So the growth of Drosophila will be reduced twice in this condition. And also because Drosophila is very small, and then we need to perform injection, and so also sometimes you need to choose which one man and which one male and which one female. You need to see using microscope. But if you become expert using Drosophila, without microscope, you can differentiate which one male and female. And also, I think it is also good to have qPCR, I think, because qPCR is very important to measure molecules that involve in our study. Also, I think we can also establish how to perform DNA extraction, RNA extraction, because when we deal to qPCR, we need to prepare samples until becoming DNA. However, I see maybe I know from the information from my friend that we also have qPCR in medical faculty, so maybe we can collaborate with them. So I think that's all, Sony. The main equipments that we need to establish a Drosophila facility in our faculty.
Thank you, Dr. Emsali. Maybe we need some equipment like qPCR or specific microscope to do the research about the Drosophila melanogaster. Maybe hopefully we can get a lot of equipment to fulfill the research about Drosophila melanogaster in our faculty. Okay, the next questions from Mrs. Hapsha Harina. How are monalizin as toxin have advantages in technology pharmacy? And are there synthesis monalizin is if maybe if not enough, yes. There are the other there is are there any method to get the monalizin if not enough to do in the research? Please, Dr. Emil Salim.
Okay. How monalizin have advantages in technological pharmacy? So what we can say about the advantages of monalizin, about the molecular mechanism. So this is, I think, not related to technology pharmacy. This is about the science. But from the science, we can provide the insights that can be used by technology to improve the technology. So first, we can learn about how the mechanism of action of toxin and host immunity. Then we can provide or can get the mechanism, and then from that mechanism, we can modify that mechanism to reduce or to increase the immune function. However, we can use for controlling pests. So this is maybe related to the biopesticides. So we can develop this toxin to kill pests like mosquito, and also the pests that can come or can make the fruits broken. So we can develop this toxin to become biopesticides. And also we can develop this toxin for biosensors, because this toxin have a specific diameter. So with this specific diameter, as you know, different molecules have different size. So if this diameter, if this toxin, if we introduce to certain molecule, and then if this toxin is blocked, so we can say that the size of this molecule is this kind of size. So example like that. So this POR already used in the UK to sequence amino acids also, because different amino acid bases will block the different pore, and also can induce different current. Okay, I think that's all what I can say about this first question. And then next question, if the monalizin is not enough, so maybe we can try both endogenous monalizin that we purify, and also we can produce recombinant monalizin as well, and then compare the effect, which one better, and then we can compare which one is more efficient to be produced, and then which one is more cheap, which one is cheaper, then we can use that one. So that is my answer for Habasha Rina.
Thank you, Dr. Emil Salim. Maybe it means that the new resource or the new method to get the monalizin from recombinant monalizin maybe become a new project for our research. Okay, the next questions from Mrs. Hayatun Nufus. Yes, first, thank you very much, Dr. Emil Salim, for your outstanding presentation. If I'm not mistaken, only toll and check pathway were involved. Does this mean something? What happened if all pathway involved? Is there any specific difference in the result if another pathway involved? Okay, please, Dr. Emil Salim, maybe it's about the mechanism of action of monalizin toxin.
Oh, thank you. Nice question, Hayatun Nufus. Yes, you are right that the pathway that involved in the host is toll and JAK pathway. So yes, of course, this means something, because this is toll is the pathway that only sends gram positive and fungi. However, like IMD pathway, which is not involved, so that is sense for gram negative bacteria. So as we can see, and as I presented before, so actually toll pathway detect gram positive, but Pseudomonas entomophila is not gram positive, that is gram negative. So this is new. So maybe this pathway not detect the bacteria directly. However, maybe Pseudomonas entomophila induce the necrosis, and then after necrosis is performed and occurred, then this necrosis maybe induce the release many molecules. Then this molecules is detected by the toll pathway. So this is new finding also. And then JAK pathway. This is JAK pathway, also the pathway that can induce by bacteria, and also JAK pathway, JAK-STAT pathway can be induced by the stress such as temperatures. So if this is involved, so we can say that this pathway is important. So in the future, if we want to manage how to increase the protection, if we attack by this kind of monalizin, we can induce this pathway by specific known molecules, or we can find molecules that can specifically induce this pathway. Then this pathway will attack that bacteria or protect us. And also there are JNK pathway. JNK pathway is the pathway involved in also detecting bacteria, and also the pathway needed for repair or wound healing. So also if this pathway involved, we can also manage this pathway to increase, to protect us from this bacteria. So yeah, so this is what I can say about the involvement of this pathway. So then the most important, if we know the pathway, we know how to protect us from this bacteria. So this is how scientists work, finding pathways to know which step they can block or which step they can induce to protect or to kill the intruders. Is there any specific difference in the result if another way involved? Yes, I already mentioned different pathways, also not only involved in killing the bacteria, also they involve in different mechanism like wound healing, etc.
Okay,
This
thanks.
is my answer from Ayatun Nufus question.
Thank you, Dr. Emil Salim. Wow, it's actually it's a unique and a new information for us, and I think it's a good new project for us to identification to develop about the research. And now we know that the from the mechanism we know which one of bacteria will be killed by the monolysine toxin, and then we can use it. We can applicate in the surface health better. Okay, the next questions from Mrs. Anayanti Arianto, our lecturer in Faculty of Pharmacy. Thank you to Dr. Emil Salim for your good presentation. I want to ask you about how the mechanism of this pore-forming toxin or monolysine damage the Drosophila gut. Maybe it's a new an important information for us about the monolysine from Drosophila, but monolysine can damage the Drosophila gut. Please, Dr. Emil Salim.
Okay, thank you, Professor, for these interesting questions. I was asked this question many times. Okay, however, I would like to answer this question here because this is also the first time question in Indonesia. Okay, for how monolysine damage the cells in the Drosophila gut. So monolysine make pore and then make cells become leakage. Then the cells become broken. However, how the mechanism? So the mechanism in this toxin is not clearly elucidated. Yeah, but we can say our speculation how this mechanism involved. First, I would like to say again, there are other mechanisms how this toxin make pore. So first, bacteria will release monomer of this toxin, only one. And then this monomer will bind to the target cells. And then when this monomer bind to the receptors in the target cells, this monomer with other monomers will form polymers. And then these polymers will make pores in the cells and then make cells become leakage. And then cells broken and then the organ of that cells side will be broken as well. So however, in our cases, we make artificial bilayer membrane. We make by ourselves this bilayer membrane. Of course, this bilayer membrane don't contain receptors. However, this monolysine can make pore. So maybe the mechanism of action is the interaction lipid to lipid. So because this, I mean maybe the interaction is between monolysine and the lipid. So maybe monolysine have a certain structure that can bind to the lipid membrane. Then after the interaction, the pore is formed. And also we found that the RNA synthesis or translation mechanism that can translate from gene to protein is reduced by this monolysine. However, the exact mechanism from the first to the end is not known. So this is also what we need to study in the future. So thank you, Professor. So that's all what I can say about the mechanism of action of this protein or toxin.
Thank you very much for the good answer from Dr. Emil Salim. Maybe are there any questions from the participants? Okay, maybe we still have 5 minutes more in this discussion. So I would like to ask Dr. Emil Salim. We know that we can use the high-speed AFM in movement protein of monolysine. But I would like to know, could we calculate the how many active monolysine from the high-speed AFM.
Oh, how active?
Yeah, how could we calculate how much the active monolysine from high-speed AFM?
Okay. So I think using HSFM, what we can do best is to observe the movement of the protein. So if you want to see the how active, so maybe only what we can is compare this monolysine to other protein. So we can see how fast this protein can make pore or can attach to the membrane. So that is, for example, so only comparing how long this monolysine make or attach to the membrane. So what we can do best using this equipment is to observe the dynamic action of molecules. So I think that's all what I can answer.
Okay, Dr. Ahmad Salim, maybe we can back to the first topic about the Pseudomonas entomophila. Is the Pseudomonas entomophila the only one of the bacteria in Drosophila melanogaster, or there are any bacteria in Drosophila melanogaster who which can produce the monolysine?
Okay. So Pseudomonas entomophila is the only bacteria that can produce monolysine that can attack Drosophila. So this is research is very important, the invention of Pseudomonas entomophila, because we need to perform the experiments, or we need to find the mechanism of action, how Drosophila melanogaster deal with the attack of pathogen. So then the researchers before us find what is the pathogen for Drosophila melanogaster, and then they found this Pseudomonas entomophila. And then recently we only found that this bacteria is the only bacteria that can produce monolysine.
Okay, thank you very much, Dr. Emil Salim. Maybe we have got much more of information and new information from Dr. Emil Salim, especially about the monolysine and from the powerful organism that is Drosophila melanogaster. Okay, to all the participants, thank you very much to Dr. Emil Salim as a keynote speaker today. And
thank you,
to
Mrs. Ade.
Yes, and to all the participants who have been paying attention with enthusiasm. And we have got a lot of insightful about the molecular and functional analysis of monolysine. And hopefully we have a good understanding. We have a good understanding and improve our capacity, especially in research and review article about the powerful organism. And then we thank you very much for your kind attentions, and never get bored to learn. Maybe if I could conclude our speech today, that the Drosophila melanogaster is a powerful organism, so that and it's also conclude the scientific research that it's so cheap and easy to use, and also 75% of human disease genome is same with the Drosophila melanogaster. Okay, thank you very much. Finally, thank you, and never get bored to learn. And I would like to hand this event back to the master of ceremony. Thank you very much, Dr. Emil Salim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, thank you so much, Mrs. Ade. We just got a bunch of knowledge from the lecture. Thanks to our amazing speaker and moderator who broke this all for us. May all of us get the benefits from the lecture. To express our gratitude, we would like to award a certificate to our speaker and our moderator. First, we would like to invite the Honorable Dr. Emil Salim. Certificate of appreciation is awarded to Dr. Emil Salim as the keynote speaker. Amazing presentation and knowledge you just shared.
Thank you very much.
Well, doctor, we will take a picture for a while. So please kindly give your best smile. Okay, 1, 2, 3. Okay, thank you, doctor.
Thank you.
And we would like to invite Mrs. Adesri Rohani. Certificate of appreciation is awarded to Mrs. Adesri Rohani as a moderator.
Thank you very much.
Thank you, Mrs. Ade, for your willingness to guide for our session. Okay, please kindly give your best smile because we would like to take documentation. Okay, 1, 2, 3. Thank you, Mrs. Ade.
Thank you very much.
Ladies and gentlemen, well, we are going to the next agenda, the parallel oral and poster presentation session. In this session, all of candidate competition would be divided into 4 rooms by the committee. Please wait until the committee invited you to the room. And all of participants can join one of the rooms in breakout room. The committee will help you to join one of the rooms. Please wait until the committee approve you to the room. Thank you.
Bisa serta.
Ladies and gentlemen, the parallel oral and poster presentation session have been done. All of candidate competition and participant could take a rest until 1:30 p.m. We would you join to main room at about 1:30 p.m. for awarding and closing ceremony. Thank you for attention. Ladies and gentlemen, please your attention. The committee are waiting for the decision of the judge. Please wait a moment. Thank you.
Hi, welcome to Medan. Medan is the capital of North Sumatra province and also is the third largest city in Indonesia.
Medan, mantap kali bang.
Merdeka Field and Tirtanadi Water Tower is an icon of Medan city built by the Dutch government. Medan has a favorite tourist spot such as the Great Mosque Al Maksoum and Sisingamangaraja Street. Istana Maimun, the royal palace of Sultan Delhi, built by Sultan Mahmud al-Rashid since 130 years ago, located at Brejan Katamso Street. There are few places of religious tourism in Medan like Maria Anai Velangkani Shrine, Emmanuel Church, which is one of the oldest church in Medan. Chong Afi Mansion, which is a building with a strong history and culture of Chinese architecture. Medan has the oldest hotel named Hotel Ina Dharmade
pancur sisada melameringis mo bagai turang alemar di dakalakmo bekel masada duan dando aleka di papo duan dando aleka di papo.
Ketemu
pancur sisa melameringismo bagai turang ale merdida kalakmo bekel masada duan dando duan dando duan dando ale kapitapo duan dando duan dando duan dando ale kapitapo.
Ladies and gentlemen, we are begin so this event about a moment. Now please your attention. Ladies and gentlemen, welcome to our event. Thank you for wait a moment. Dear ladies and gentlemen, now we are at the awarding session. As previously mentioned by the committee, there are 4 award categories. There are the best of oral research presentation, oral review presentation, poster research presentation, and oral review presentation. And now we have already got a list of the winners by the judge. I'll read the name of the best. Ladies and gentlemen, first is the categories. The best of poster research presentations. There are no the winners in these categories due to there are no participants in these categories. So we are going to the best of poster review presentations. There are 3 winners of these categories. I'll read the name of the third winner first. And the third winner first of poster review presentation is Intan Faradiba with the presentation title is In Vivo and In Silico Fracture Healing Research Models, a Review. And the second winner is Savannah Ersa with the title poster is The Effectiveness of Salvador Persica Miswak on Periodontal Health Literature Review. And we are going to hear the first winners of poster review presentation. And the winners of poster review presentation is Hayato Novus with the title poster The Most Potential Compound of Zingiber officinale in the Treatment of SARS-CoV-2, a mini review. Congratulations to all the winners. Okay, ladies and gentlemen, we are going to the winners of oral review presentation. I'll read the name from the third winner first. And the third winners of oral review presentation is Mr. Roni Abdi Saputra with the title The Role of Flavonoids as Cardioprotective Agents, Doxorubicin, a Review. And the second winner of oral review presentation is Mr. Fauji Satria with the title The Possible Role of Plant-Derived Essential Oils Against Fatigue in Post-Acute Sequelae COVID-19, a literature review based on evidence of essential oils on fatigue. Now, we are going to the first winner of oral review presentation. And the first winner is Mrs. Imelda Ferrandina with the title The Level of Patient Satisfaction on Pharmaceutical Service in Indonesia, a Review. Congratulations to all the winners. Now, we are going to the last of categories. And I'll read the winner of oral research presentation from the second place. The runner-up of oral research presentation is Dea Nur Fadilah with the title Immunomodulatory Effects of Cermai Leaf, Pilanthus acidus kills ethanol extract on normal male rats and cyclophosphamide induction. And the first winner of oral research presentation is Mrs. Rima Elvitra Rambe with the title The Implementation of Clinical Pharmacy Service in Hospital at North Sumatra. Congratulations to all the winners. To all of the winners, I would to invite you because we will to take a documentation. Please sit in a good position and please kindly give your best smile. To all of the winners, please turn on your video first. To all the winners, we want to invite you because the committee will take the documentation. And we want to invite Dean of Faculty of Pharmacy and the committee to take a documentation. Please turn on your video. Okay, the first we will take a documentation with all of the winners. So all of the winners, please turn on your video. Please wait a moment. The committee will take a documentation. To all of the winners, please sit up on a best position. Committee will take the documentation. Kindly give your best smile. One, two, three. Okay, thank you to all the winners and congratulations. Now we want to invite the judges because we will to take a documentation with all of the winners. To the judges, please turn on your video. The chairwoman of committee, please turn on your video. Thank you. So all of the judges and all of the winners and the chairwoman committee, please turn on your video. I'm so sorry for the technical problems in here. And now we will take documentation. Please kindly give your best smile and 1, 2, 3. Okay, thank you for all the winners, the judges, and the chairwoman committee. So all of the winners, congratulations. And we will give some information. Certificates and awards will be given in the main event of the Desnatalis Faculty of Pharmacy, Universitas Sumatera Utara on next Saturday, 20 November 2021. Dear ladies and gentlemen, we are now at the end of the event. As the closing, we have a closing remarks which will be okay. We are so sorry because there are any problem in here. Dear ladies and gentlemen, unfortunately, at the end of awarding best presentation. So this is the end of our event. I would like to close it with a quote by Albert Einstein. Once you stop running, you start dying. The world will not be destroyed by those who do evil, but by those who watch them without doing nothing. So that we hope all of us, being a person who's never bored, to keep learning and keep trying to do anything that will give a good contribute to our faculty, our university, and our country. May all of us gain a million knowledge here and also make us curious to gain even more. Dear ladies and gentlemen, last but not least, on behalf of the committee, we would like to thank you so much for your attention and please apologize if there are mistakes or inconveniences during this event. See you on the next research and review article competition. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam
wabarakatuh.
warahmatullahi wabarakatuh.
Congratulations to all the winners.
Thank you.